Hello, everyone, and welcome to today's webinar, Troubleshooting with Quality Control Data in Medical Laboratories for Beginners. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by LGC Clinical Diagnostics. To learn more, visit www.lgcclinicaldiagnostics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box to the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Oswald Sonntag, Independent Consultant. For a complete biography on our speaker, please visit the Biography tab on the left of the screen. You will not find that his name does mean Sunday, so that is a fun fact that you will not find on his Biography tab. But Oswald, you may go ahead and begin your presentation.
Okay, thank you, Cassie. Dear attendees, my dear QC friends, I'm happy to present you today troubleshooting with quality control data in the medical laboratory. Firsthand, I like to thank LGC, Technopass, and LabRoots that they invited me to give this talk. Quality is not an act, it is a habit. So that's already known in the early days of our life. I give you a short intro and maybe a little smile for you. The introduction is my early days in the lab. As you can see here on the left side, my boss, the director of the lab at that time, was invited for an important conference. And May, as a young assistant, did the measurement. And as you can see here on the screen, I did a method comparison, a new method versus a routine method. And there is a single point which falls outside of the comparison. So usually nobody will ask on the line points which are on the line, but everybody is fascinated by this single point. Sometimes people decide and call this an outlier. They delete it and took it away, just not being asked for this different result. I got fascinated by this situation, and I dedicated finally my life in science to this single point. I want to find out what is the reason, what is the error, and how can I solve this error. So the key to quality in the lab is you. You have to deal with patient samples, the analyzer, and the reagent, and you have to take care that all behave the way you wish or the regulations are recommending. What is quality control in a medical laboratory? I'm just repeating a little bit from my last webinar for those who didn't have the chance to look at it. Laboratory quality control is all the measures put in place to eliminate the risk of non-conforming outcomes. It involves systems that safeguard the accuracy, reliability, and timeliness of lab results by ensuring the early detection of results or measurement errors and the procedures to correct them. This finally contributes to the reliability of laboratory results and also to patient safety. How much of an error would you accept if you see a doctor? So for me, clearly it's 0. And for you? Consider that laboratory test results have always a little what we call uncertainty. So you know if you do a measurement twice that the values might be slightly different. So that's something we have to live and deal with it, and that's why we have quality control. So in medical diagnosis as well as in laboratory medicine, the acceptable error or tolerance for errors is remarkably low. Ideally, the goal is zero error whenever possible. But it is important to understand that absolute error-free diagnosis is not always achievable due to numerous factors such as the complexity of medical conditions, the limitations of medical tests, and the inherent variability in human biology. Why do we need quality control procedures in the medical laboratory? Those procedures in the medical laboratory are essential to ensure patient safety, to maintain compliance with regulations, prevent errors, and provide accurate and reliable test results. They are the basis of responsible and active healthcare delivery and research as well. Now I'm talking about troubleshooting in the medical laboratory. See a little jokey part here in this slide. Troubleshooting involves a systematic approach to detecting and resolving issues that show up during the quality control process, especially when quality control results fall outside defined acceptable limits. Troubleshooting aims to ensure the accuracy, reliability, and validity of laboratory test results. There is a document provided by the Clinical and Laboratory Standard Institute that's called C24. And within this document, there are different steps for doing proper troubleshooting, detecting the errors in the analytical process, stop reporting patient results if they are outside the limits, in the limits of the QC, investigation to determine the root cause of the error or errors, implement corrective actions, evaluate the impact on previous reported results, and mitigate, finally, patient risk, implement corrective action to avoid reoccurrence of errors. So in laboratory medicine, we are using the Levi-Jennings chart, and I'm using the chart and interpretation of the chart today in order to discuss some problems you might find in your laboratory and how to solve those problems. So it is important to record every quality control value, including those that are out of control. The object of quality control is not to produce a beautiful control chart, but trying to keep all results within the 2 or 3 standard deviation ranges. You have to recognize that already 5% of control values are expected to be out of the 2 standard deviation ranges. This is because of the normal distribution of values. We will see a graph very soon. 2 standard deviations is considered as a warning limit. Take in consideration if quality samples are routinely rerun until they fall within the current control limits and the outliers are not recorded, the acceptable range will become small each time they are recalculated. So eventually they will approach 0 standard deviation and become useless and unattainable. I will show you an example later. So this is the graphical presentation, which we call Levi-Jennings chart. It's interpretation of quality control data, which both are graphical and statistical methods inside with this graph. Quality control data is most easily visualized by this chart. And here you see on the left side the bell-shaped curve, the normal distribution curve, or the Gaussian curve, whatever you want to call it. That's the curve which shows a normal distribution. And in the center of the slide, you see the Levi-Jennings chart. The green line is the mean value. The yellow line, the 2 standard deviation plus and minus, and the red line, the 3 standard deviation plus and minus. we are using this graph to show our quality control data either by reporting our daily results or the measurements we did. So this is a useless and unrealistic situation, but I want to show this. Everybody would be fascinated if you have a method which runs like this, that all the measurement points of the quality control material is hitting the target or the mean value. But this is unrealistic situation, and therefore it's useless. So the real situation might be like this. You see that we have here 5 quality control measurements which are around the mean value within the 2 standard deviation range. This is fine, and we can accept to release patient results to the ward or to the clinician to be discussed and reported to the patient for treatment or diagnosis. Now we have a situation where a single point hits the 2SD range. Sometimes people get nervous, sometimes people decide to not report patient results, but as I said in my previous slide, this is just a warning that you have to take care what's happened next. Usually under this condition, you are allowed to report patient results. You can draw a line between the points, which gives you maybe a better impression about what happened. Here you can see it's moving downwards. See what happened next. So how to deal with the 2 standard deviation rules? The 2SD rule is only a warning rule that is violated when a quality control result falls outside the 2SD limits. Take into consideration that approximately 5% of all quality control material results may fall between the 2 and 3SD limits. So the 2SD rule only warns that random or systematic errors may occur in the test system. Patient results can be reported in this case. Remark, using the 2SD rule as the rejection rule results in about 5% false rejection, creates unnecessary extra work, and may delay reporting of patient results. Cost increase as well. So now we are focusing on the situation having measurement which hits the 3SD limit. You can see the arrow and the black point on the Levi-Jennings chart, and here we might have an issue and a problem, and we have to do then a troubleshooting. That's the topic of today. Just for curiosity, the point is not always on the low side, could be also outside, up or down. So this is just an example which I'm using to show you how the situation might be. So here we clearly have a value which is outside the 3SD range. And we have to see what we have to do in this case. When an analytical run is rejected because quality control is out of acceptable limits, in our case the 3SD range, it is often necessary to determine if patient results reported between the last acceptable run and the rejected run needed to be repeated. This decision should be based on the nature and the size of the error. So, for example, a 5% bias may not have heavy clinical significance for most patients, but a 25% bias is unacceptable for nearly all patients. Biases in between these extremes need to be examined on a case-by-case basis. So my recommendation here that the lab management should make decisions at which limits you have to work, or there are countries like the country I'm working in is Germany, where the government has set those limits and those regulations. If analytical errors have been clinically significant, then some, if not all, of the patient samples should be retested. So don't report any patient results in that case, starting with the samples analyzed just before the rejected control samples. So what you have to do in this case, have you just calibrated the system? Could be this is the reason. Have you used a new lot of calibrator or a new bottle of reagent? Has the lot changed of the reagent? Have you used a new bottle of the quality control material or a new lot of the quality control material? So that's something which you have to check prior to any further investigation. So the review points of failure put here together some issues, some points, some topics which we have to consider. So firsthand, check if there is a human error. Have you ever read the instruction of use? I know it from myself. If I buy a new device, I try always to play with the device without reading the instruction. But then if I'm struggling, yes, then I'm referring the instruction of use. And it's better to read the instruction prior to start with the process, with the adjustment of an instrument, operating of the instrument, preparing a QC material, and so forth, so on. So there might be an instrument failure. Check if there is an error message. This might be printed or displayed on the instrument. Refer to the troubleshooting section of the instrument manual to figure out what the reason for this error message could be. Reagents. Have you used the correct reagent? Check the lot number. Is there sufficient volume of the reagent in the bottle? What about the shelf expiry date in the refrigerator and also on board? Or is there incorrect storage that, for example, the reagent has left at room temperature for quite a long time, which may have deteriorate the reagent? The calibrators. Have you used the correct calibrator, which is recommended by the manufacturer of the reagent and instrument? Check the lot number, the assigned values, the preparation, the storage conditions, and also the expiry date. Inspect the calibration trace of last calibration. Quality control material. Have you used the correct material, the right lot number? Check the assigned value, the expiry date, and in-use expiry date. Storage condition as well. Preparation. Prepare it fresh or also allow enough time to dissolve, or if you take it out of the freezer or refrigerator, wait till the QC material is in the proper condition to be used on the instrument. Here are some additional and helpful investigation tools. I put some information together on this slide to help you what you can check for in the case if you have a QC problem. So the manufacturer, have you used the right product, the set from the right manufacturer? What a product name, is it correct? The fluid type you're using is a lyophilized material, liquid material, the lot number, the onboard stability of both reagent and maybe also the QC material, the open vial stability, refer to the quality control product instruction for use, abbreviated IFU. If you use the right fill volume of both the reconstitution fluid or for the sample volume in the cup on the analyzer, the type of the quality control material using assayed or unassayed material, and also check the levels and the lot numbers, expiration, quality control, sample identification code, and also catalog numbers and the methods in use so that they fit together. Now I'm using the graphical presentation, the Levi-Jennings chart, in order to discuss some situations which may happen. This is a situation of a shift. A shift is a sudden change of values from one level of the control chart to another. You can see it here on the right side, that the first 4 points moving down, then there is a shift upwards, and you are measuring higher concentrations in those QC material. Another situation might be a trend. A trend is a continuous movement of values in one direction over 6 or more analytical runs. Trends can start on one side of the mean and move across it, or can occur entirely on one side of the mean. Here in the graph you see that the values are moving downwards and reaching the 3SD limit. Here you have to find out the reasons, and I put together some information for you to make troubleshooting a bit easier. When trend or shift is identified, the following corrective actions can be taken. Check the expiration date of the reagent. Check the expiration date also of the control material you are using. If a new control material was used, make sure it was properly prepared. Retest the control material. And important, do not report patient results if you are outside of the limits. Documentation of flags and corrective actions. Every laboratory should have a logbook, electronic file, or whatever, for the quality control results, where all events and actions are recorded. This is particularly important for those errors that are observed and be troubleshooted. If the same error can be fixed with a particular corrective action, this log may help you later on to speed through the troubleshooting process. If multiple errors of the same type are occurring, this log is also helpful to identify the long-term trends and issues with the performance. It is both a regulatory mandate and also best practices to maintain a history of the instrument behavior. Troubleshooting if problems occur. There's always the recommendation to rerun the quality control sample. This, for me, is if you don't know the reason and you're not sure if you have used the proper control material. But if you rerun just the same QC material, quality control material, you might get exactly the same value. So if you repeat the value and it's still out of the range, you may run a new vial of quality control material. If the new control value is within the acceptable limits, so within the 3SD range, record the values and proceed with testing of the patient samples. If the new control value is out of control, outside of 3SD, do troubleshoot of the instrument, checking the sample, reagent delivery, mixing, lamp integrity, reaction temperature, and last but not least, if there is a change of the reagent lot number. The last step I would recommend is to do a recalibration of the method. Because if you calibrate and there is an error on the instrument, on the reagent, or the combination of both, you may calibrate this error away, and it may come back later in the month or the week. So be careful and have a good reason while you do calibration or recalibration of your method. Talking now about the reagent lot-to-lot variation. If control values shift after a new reagent lot number has been introduced, then rerun some patient samples with normal and abnormal values. If those values from patient samples correlates good, control value shift are probably acceptable. If they are poor, you have to consider that the reagent may be bad. Try a new lot number of the reagent. And to be fair with the manufacturer, if you have such a problem, which you corrected by using a different lot number. Please inform the manufacturer that this happened so that they can inform others about this problem. I've seen this literature, this publication from van Schorenstein, which was published last year. And here it's nice to see on the left side how you zoom up with your performance. So if you look at the within day performance, it seems to be quite stable in this situation. So there is only a minor variation between the values. If you look the one step down, day-to-day variation, you see that's more amplitude behavior of the material. Then if you do calibration, see that has a more stronger impact to the performance of your instrument and the recovery. And also if you do a lot change without doing calibration, this has an impact. And this end up in the total variation at the end. If you combine all these variations together, here you see the final total variation on the left side. On the right side, that's interesting to see behavior of a QC material and a patient sample while you do lot changes. Also here on the y-axis, the concentration, on the x-axis, the time, and you see the dotted line here, that's the lot change, another lot change, another lot change, and here the fourth lot change. And you see that the QC, that's the blue ones, show clearly that there is a difference between lot to lot. If you just do check with your patient material, and this is always the same patient, you see it behaves same or similar. If you do measurement of more patient samples or more often the patient samples measurement, you can use this also as a type of additional quality control checking. Common causes for random errors, the operator issue. I'm finger pointing again to this human error and human behavior, as my advice is please read the instruction carefully and refer to the instruction to better understand why failures or errors may happen. The reagent preparation error in or inconsistent preparation of the reagent. These days, most of the reagents are ready to use, but still there might be consider that you are using the reagents at a correct temperature. Failure to consistently power fluctuation of your instrument, temperature fluctuation, either in the environment or the incubation chamber of the instrument. The technique, you might have a very sensitive technique for your testing. Quality control material, yes, also the quality control material may have issues. For example, if you are not preparing them well. So my recommendation is use fluid, ready to use quality control material to avoid any dilution or problems in the reconstitution of your QC material. There might be also software malfunction and also contamination. Common causes for systematic errors, that's the calibration. This is a primary cause of systematic measurement errors in the test system. So please take most of your action to have a proper calibration and using the right calibration material, following again the instruction of the manufacturer of the reagent. So reagent integrity, new lot or newly prepared, they may behave different. Check again always the lot number, even if you use it for a while, check it if it has moved or changed. Second most common cause of the systematic errors is the reagent lot to lot situation. Lack of or inconsistent instrument maintenance, please follow the instruction of the instrument manufacturer and follow the maintenance properly. Failure to follow manufacturer's instruction for performing the test could be a problem for systematic errors. Levi-Jennings chart that is improperly set, if you have chosen the wrong target values, the wrong mean, the standard deviation not properly calculated, or you remember the beautiful looking Levi-Jennings chart, which does not allow any error because it's so narrow and all the values are on the target. Instrument malfunction, reagent or sample pipetting malfunction could be an issue for systematic errors. The light source, incubation chamber temperature may have shift the temperature. Operator technique or technique sensitive test, software malfunction. There might be recent updates which may be a reason for causing a problem, and possible contamination of the reagent, the tubing, the water flushes, and ancillary equipment such as pipettes, so on. Now, there is another tool which I also like to recommend you by doing troubleshooting. A very helpful tool is using peer group data. A peer group in quality control is a group of medical laboratories that perform same or similar testing procedures. They may use the same instruments, the same reagents, and they share their quality control data with each other. This is the software tool. The primary purpose of a peer group is to monitor and maintain the accuracy and reliability of medical laboratory test results. Purpose of peer group and quality control, comparison and benchmarking. Laboratories within a peer group compare their own quality control data with that of other labs using the same instrumentation, and see if they can identify variations and deviations from the established quality control criteria. Identify trends by analyzing data collectively. Peer groups can identify trends in quality control data, such as shifts and drifts, that may indicate potential issues with your test methods, instruments, or reagents, especially if there is a lot change. And you can use it for troubleshooting. Peer groups serve also as a valuable resource for troubleshooting when unexpected quality control deviations occur. So labs can consult with other members who may have experienced similar issues, and maybe they have found solutions helping you on your way to fix that issue. Using peer group data for troubleshooting, data comparison. When you encounter a quality control deviation in your laboratory, quality control data, you may compare your results with those of other laboratories. So that's the beauty of the peer group. See how the others are performing. If multiple labs are experiencing the same problem, it may indicate a broader issue with a test method, reagent, or instrument. Sharing the knowledge, reach out to your peers with the group, within the group to discuss the problem. They may have faced similar challenges and can share their experiences and solutions. So this can save time for you and also resources to identify the root cause of your out of control situation. Collaborative problem solving, collaborate with other labs in the peer group to troubleshoot the problem. This can involve sharing protocols, procedures, or adjustment strategies that have worked for others. In some cases, you may even visit their laboratory to observe their processes firsthand. Feedback and continuous improvement. Use the feedback from your peer group to implement improvements in your laboratory process in order to avoid QC problems and report reliable patient results. Regulatory reviewing and discussing quality control data with the peers can lead to enhanced quality control practices. So that's an additional tool using the peer group data. So in summary, a peer group in quality control in a medical laboratory serves as a collaborative network for laboratories to share data, insights, and offer solutions related to the quality control issues. It helps identifying and troubleshoot problems, ensures the accuracy of test results, and promotes continuous improvement in laboratory practices. Peer group participation is, for me, therefore, an essential component of quality assurance in medical testing. Now, after I presented you some ideas about how to fix the problem, and now we have solved the problem, we have fixed it, the error is known, the error is under control, what we have to do now? Run quality control material for evidence that problem has been solved. Document what you have done in your troubleshooting logs, your quality control notes. This is important if you be faced with the same issue later on. If you have documented, you may have already a solution for it. Address patient results from previous good quality control to when quality control failure occurred. Repeat testing if needed. Exclude or inactivate failed quality control measurements from data analysis only if cause of the outlier is clearly identified. So this have to be taken in consideration. If you clearly know what the error is and how you fix that error, then you're allowed to delete these quality control measurements and don't use it for calculation of your Levi-Jennings chart limits. So now my conclusion: use statistical data and available peer group data to evaluate your out of control situation. Do not automatically rerun quality control material or recalibrate the assay. So if you don't know the reason, you just rerun the QC material. It may deliver the same result, doesn't help you to fix the problem, and you have to see what is the error and fix the error first, the problem first. Same is true with the calibration. Type of quality control failure and quality control charts using the Levi-Jennings charts may give a clue to the nature of the problem. So values are too high, too low. So that gives you an idea in which direction you may have to do the investigation. Have a structured approach to troubleshooting. Have a process for managing your patient results. This is usually also given by the management that if you're outside of your QC limits, that you're not reporting patient results. Manage introduction of new lots to the laboratory, especially those of the reagents. They are causing more often problems in your daily QC. As usual, prevention is better than cure, and ideal situation is do it right the first time. So I'm at the end of my presentation. If you want to get more knowledge about the basic application in clinical laboratory quality control, there are two booklets available which you can request by the link which is given here. One is just a basic and the other booklet is on advanced application. This is for training purposes. Thank you for your attention, and I'm now available for questions and answers. I like to thank again Laboratories for moderating me in this webinar, and also LGC Technopass for making this webinar possible. Thank you.
Thank you, Oswald, for your informative presentation. We will now go ahead and start the live Q&A portion of the webinar. If you have questions you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit, and we'll answer as many questions as we have time for. And Oswald, you can go ahead and stop sharing your screen at this point.
Okay, yeah.
Thank you.
Thank you, Kathy.
Of course. So I'll go ahead and ask our first question here. Why do labs insist on using a third-party QC, and what are the pros and cons?
Usually, I have to tell a bit more story around this. So usually companies are optimizing their reagent calibrators and QC material and offer this as the proper solution for the medical laboratories to check the performance of your instrument. If you use an independent or third-party control, you don't have this biased issue that you have an optimization between the reagent and the QC material or the calibration. So with the help of independent QC material, very often much easier to detect issues and problems with your instrument or the combination of instrument and reagent. So therefore, it's highly recommended to use independent QC material. I have to add also a little bit discussion that sometimes the manufacturer of the reagent and instrument is recommending to use their QC material because if you have issues with this combination, that they rely only on their QC material. But if you can show with patient results and also with the help of independent QC material that there is an issue with a reagent or instrument or combination, that's very helpful in the discussion. And additionally, you have to take in consideration that the so-called proficiency testing or ring trials are also a tool of independent QC material. Unfortunately, proficiency testings are running not that often. So in some countries, only 4 times a year, or in other countries, once a month. But to be on the safe side and deliver patient safety, you should take in consideration to use independent QC material. And by the way, for those labs who are accredited, there is also recommendation within the 15189 and the ISO document, which highly recommends to use independent QC material. You see, there is a lot of advantages.
Definitely sounds like there are. Thank you so much. Our next question that we have is, let's see. Can you advise the best sources for allowable error or range for chemistry and immunoassay tests?
Yeah, this is a challenging question. I can answer it the easy way that, for example, in countries like Germany, there are given limits by the government. So you clearly can use those in the German laboratories. But if you look outside, there are clear recommendations. There are recommendations from different societies. There's also the idea to use total allowable error based on biological variation. And for me, it's the management of the laboratory. They have to make a decision which they want to choose. So there could be a visit of the website, the web and homepage of the Westguard QC to find those limits, and you may be using those. You have to find a source for it. Finally, if you don't want to follow those regulations, or you don't need to follow those regulations in your country, you have to set those limits by yourself, by the management. Together with the management, they should tell you, or together with the management, you should create those values which you are using to set your limits.
Thank you. All right, we have a ton of questions coming in, so I'm just going to jump to the next one here. The next question says, independent quality control, or IQC, running high after calibration. Manufacturer controls running within range. What do we do in this case? We used another reagent with different lot, same thing. We used another calibrator, yet the independent control was still very high. What do you advise?
Yeah, there are different issues. A helpful tool can be also to look at your patient results. So sometimes laboratory are calculating the average of means, that is calculating the values of one analyte over the whole day, and calculate a mean and compare the mean with the previous day. So this is an additional tool. It's not so easy because there are no limits given by any organization, but it gives you an idea if you have a shift or trend or whatever in your QC procedure. Now going back to the original question, you do the calibration, the QC of the manufacturer is within the limits, so usually you shouldn't have any issue to call the manufacturer or to be upset about the situation. Now you measure the independent QC, and the independent QC shows you a different value. So there is something wrong. Could be also that the target value which you are using of the independent QC might be not correct, but to my experiences from the past, and it's also published in the literature, that very often due to the optimization of the calibrator, reagent, and QC material and instrument finally, you are not able to detect problems and errors properly with the so-called in-kit control. Playing with the calibrators, sometimes calibrators are linked directly to a lot number of the reagent. This is because of their optimization process, but for me, indication that there might be an issue with the reagent. Now in Europe, sorry, this I have to answer this a little bit more with giving you details. Due to the new regulation here in Europe, the so-called in vitro diagnostic regulation, there is now a request that companies, manufacturers of reagent have to report their performance of their lot. And if there is a lot change and they have a difference, they should report this to the labs so that the lab is aware. Problem there is that nobody tells what the lab has to do if there is, for example, a difference of 10%. Should you adjust your instrument? Should you adjust the calibrator or even the reference interval? That's still open, not answered yet, but under discussion. Again, the use of independent quality control helps you on your way, and you can use also proficiency testing, which shows also that you might have a problem with this combination. I'm sorry that it takes me so long to give a proper answer, but I guess that's necessary to deliver such details.
Yes, absolutely. Very important information getting out here. Okay, our next question that we have states, if the QC is made from the same material as the calibrator, how big is the risk it may not pick up issues of a true human sample?
Yeah, there's always the recommendation not to use the calibrator and the QC material produced from the same material, because the calibrator may have an issue in this composition or in the concentration. And if you use the same material in your QC material, that makes no sense. Then you can use the calibrator also as a QC material. So the QC material should be clearly different to the calibrator material. Otherwise, you can't do a proper control.
Thank you. Our next question that we have here says, What is the best practice for lab mean establishment and acceptable run criteria?
Yeah, usually you are forced to do a couple of measurements to calculate the mean and to set those limits for your laboratory. But always compare it with those which are given either by scientific societies, by the government or any other authority, or even by the lab management. Don't know if this answered the question.
Okay, thank you. We can always reach out via email if it's not answered.
Okay.
All right, our next question is, should we compare our result with the peer group if there is a trend as a first step, or should we do the corrections such as calibration, open a new vial of QC, and comparing that with peer group should be our last step?
Now, figuring out what could be the problem is time consuming and may take a lot of attention and actions. While just looking at the peer data is quite easy. If you're participating in such a peer group software program, you can easily detect by comparing your data with a peer that there is an issue. So, and if others are focused with an issue, so probably you need to have a word with the manufacturer of the reagent or instrument that there might be a problem with this particular lot which is in use. If you do an own troubleshooting, investing time and waiting for a couple of minutes, if not hours, to fix that problem, you lose valuable time to report your patient results properly and in time. So I prefer to have a first look if you have a problem and you don't know the problem. If you want to search the problem, if you want to find out what is the issue, please consider to consolidate the peer group data.
Thank you. We have another question here that asks, what are the situations when we can modify our mean and SD, e.g. shift of mean due to calibration or PM, reagent lot change, etc.?
Sorry, Cassie, can you repeat it a bit slower?
Absolutely. What are the situations when we can modify our mean and SD, e.g. shift of mean due to calibration or PM, reagent lot change, etc.?
Okay, I got the question. Thank you. Usually there are differences between lot and lot in a reagent preparation. This is because of the sources companies are using in the production. There might be variations which you have to take in consideration. In this case, you see a shift, and in the shift, you recognize now there is a difference, and you have to use the new mean for your target in the future because of the reagent. But you have to make sure that this has no impact to your patient results. So is it only with a QC material, or do you see the shift as well with patient results? Again, here, independent QC material will help you, telling you how heavy the impact is.
Okay, great, thank you. All right, our next question here asks, is the EQA process sufficient for peer comparison, or do I need to monitor peer for daily quality control?
Yeah, it's worthwhile question to recognize that the peer group data are available on a daily basis, so you can check this anytime you want, anytime you need. While with proficiency or external quality control programs, you have to wait till those programs are running and till you get your results back. Usually it's too late because my experiences, there could be 3, 4, 5, 10, even weeks between the measurements and that you get your final results for external quality control program. So peer group tells you immediately where you are and what happened to your situation if you participate with your lab with the correct instrument and reagent lot number. That's very helpful too. Additionally, again, it's using the independent QC material, which is like a proficiency testing, which you're doing every day. The difference is, for sure, the independent QC material, you have a target which is given by the manufacturer of the QC material, while with a proficiency testing, you don't have values, you just do the measurement and you have to wait till you get information if you had been successful with the testing or if you failed.
Okay, thank you. We have another one here that asks, is it safe to release patient results when the IQC fails but the manufacturer's control passed?
No, in this case, you have to figure out what is the reason. So if you don't have the reason, it's not a good practice to release the patient results. First, find out what is the reason for this difference. So if your internal QC show that there is an issue while the QC from the manufacturer behaves brave, there might be an issue. So from my point of view and my experiences, in this case, you have to find the reason for it, why there is a difference, and don't rely on just one situation. Here you have two different situations. One shows that you are in, the other shows that you are out. You have to find the reason for it. Fix it,
Okay.
and if you fix that, then probably this problem has gone. But, I need to explain you that if the manufacturer QC fits within the limits, that might be because they have a new lot. And I need to have more details of your situation in your lab. Have you changed something? Have you done a new calibration? Have you used a new reagent lot? Is there something wrong with your instrument? Have you got a flag warning from an error warning from your instrument? So that's all I have to consider prior to make a decision. But again, if you have different results, one is in, one is out, no, don't report patient results. Find the reason, fix it, and then report patient results.
Thank you. And it looks like we have time for just one more question today, so I'll go ahead and ask that. From your experience, do we see the precision and accuracy of assays improving significantly over time with improved instrument technologies, improved test methods, or do we need to be as vigilant as ever to potential errors?
That's right. The newer instruments compared to those of my early days in the lab changed completely. This is a challenge for doing quality control. As the limits get tighter and tighter and tighter, you are faced with working and focusing much more with quality control data. So there are a lot of opportunities and varieties and discussions in the field of quality, which I didn't touch today because my presentation was for beginners. So I didn't touch those complicated understanding of QC, which we need to use in the future because of a better performance of the instrument and the reagents and a better understanding of QC. So I did it the simple, easy to understanding way, but there is much more to do. That's why I recommended for those who are interested to look at the advanced training book from Technopass to get a deeper understanding of quality control. If you have the wish or if you want to get deeper, then just let me know and I can work with Technopass and Labroots for another webinar to get deeper to those details.
Thank you, Oswald. Did you have any final comments for our audience before we go?
Yeah, my comment is because I'm working since years in a laboratory, I was inventor of the QC or part of the inventing team of the QC here in Germany. My challenge and my understanding is do it right the first time. And that's easy to say, complicated to do. But if you read the instructions carefully and understand the instructions, then it makes the work much easier. And that's my words, which I would like to finish with my talk. But my very last words is to thank again Labroots, Cassie, for your moderation, asking me the questions, and LGC for sponsoring this webinar. But last but not least, thank you to all the attendees, and I'm very thankful that you spent your time listening to me and learning quality control, especially in regard to patient safety. Consider that once you could be the patient and you want to have a laboratory which delivers good quality. Thank you.
Thank you again, Oswald, for your time and your important research. We would also like to thank Labroots and our sponsor, LGC Clinical Diagnostics, for underwriting today's webcast. Before we go, I'd like to thank the audience as well for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during our on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.